Hello everyone, we are Group 8 and we are going to be talking about hyperimmunoglobulin E syndrome, HIES.
Hyperimmunoglobulin E syndrome is a rare inherited primary immunodeficiency disease with an estimated incidence of 1 in 500,000 to 100,000 individuals. In the most basic of terms, hyperimmunoglobulin E patients have hyper as an elevated IgE, which is one of the 5 most important types of antibody, and this occurs due to defects of the immune system. In normal people, the body naturally produces IgE in response to allergen and causes allergic reaction, while high amounts of IgE are also produced in response to infections by helminths or protozoa. Having greatly elevated amounts of IgE chronically causes problems with the skin, sinuses, lungs, bones, and teeth. The disease is inherited either in autosomal dominant or autosomal recessive form of inheritance. Autosomal dominant form is classified as type 1, while autosomal recessive is type 2. In 2007, a heterozygous mutation in the gene encoding the transcription factor STAT3 was found to underlie most cases of autosomal dominant inherited form of hyper-IgE syndrome, which is also known as Jobs syndrome. Furthermore, in 2009, mutation and deletion in DOCK8 gene were found to underlie the majority of the cases of autosomal recessive inherited form of hyper-IgE syndrome. Other extremely rare etiologies include TYK2 deficiency and mutation in the ZNF341 gene.
Hyper-IgE syndromes are characterized by eczema, recurrent bacterial skin and lung infections, and obviously, greatly elevated serum IgE. However, each of the two types of hyper-IgE do exhibit particular clinical manifestations which may be more severe than in the other. Type 1 hyper-IgE syndrome caused by mutation in STAT3 causes a variety of connective tissue, skeletal, and vascular abnormalities such as respiratory issues due to lung parenchyma changes and scoliosis. In addition, autosomal dominant hyper-IgE patients have a characteristic facial appearance with coarse facial features with exacerbated pore size, deep-set eyes, and broad nasal bridge. On the other hand, type 2 hyper-IgE syndrome caused by DOCK8 deficiency is characterized by severe mucocutaneous viral infections such as warts and a predisposition to malignancies at a young age. Food allergies and asthma are also much severe in DOCK8 deficient hyper-IgE8 patients as compared to STAT3 mutation hyper-IgE patients.
Let's first talk about autosomal dominant hyper-IgE. Autosomal dominant genetic disorder requires only a single copy of an abnormal gene to cause a particular disease. The abnormal gene can be inherited from either parent or can be the result of a mutated gene in the affected individual. The risk of passing the abnormal gene is 50% for each pregnancy. As mentioned earlier, it occurs due to mutations in STAT3 gene located in human chromosome 17q21 in around 70% of autosomal dominant hyper-IgE patients. However, the etiology of other 30% is still unknown. STAT3 is a major signal transducer and activator of transcription, which is involved in mediating cytokine signaling via the JAK-STAT pathway. The JAK-STAT signaling pathway is involved in processes such as immunity, cell division, cell death, and tumor formation. Patients have shown defective T helper 17 differentiation. T helper 17 cells are believed to be important in the extracellular defense against bacteria and fungi and produce interleukin 17. Cytokines upregulated by interleukin 17 include the pro-inflammatory cytokines TNF alpha and interleukin 6, amongst other cytokines and neutrophil growth factors. Both pro-inflammatory cytokines such as interleukin 6 and anti-inflammatory cytokines such as interleukin 10 signal through STAT3, which may explain how the hyper-IgE disease is of too much inflammation as seen locally in pneumonias with exuberant pus, and also too little inflammation with the relative lack of systemic signs of illness. All reports have shown that the mutations associated with autosomal hyper-IgE result in a normal amount of STAT3 protein produced, but the function of the protein is affected, resulting in a defective host defense. Mutations are largely located in the SH2 domain, which mediates protein-protein interactions, and the DNA binding domain, which mediates protein-DNA interactions. The majority of reported mutations are missense, and others are small in-frame deletions with several hotspot mutations. The pathogenesis of autosomal dominant type of hyper-IgE has been shown to be linked heavily with tissue-specific STAT3 mutations, due to which there are many characteristics of this disease. A 2007 study compared the effects of STAT3 mutation in mice, which were studied in a 2006 study, with the symptoms of hyper IgE patients and tried to provide an explanation for how the various symptoms occur. For example, one of the hypotheses was that a deficiency of STAT3 in the mouse brain is associated with increased inflammation, demyelination, and astrocytosis in response to nerve injury, which is consistent with the parenchymal brain lesions found in persons with the hyper IgE syndrome. In another suggestion, it was linked that myeloid-specific STAT3 deficiency causes increased myelopoiesis and eosinophilia in mice, and eosinophilia is a consistent feature of the hyper IgE syndrome. On the other hand, let's talk about the autosomal recessive form of hyper IgE syndrome, which is mostly, but not always, due to mutations of the dedicator of cytokinesis 8 DOCK8 gene, which is located on chromosome 9p. Since the discovery that loss of function mutations in DOCK8 underlie autosomal recessive hyper IgE in 2009, an estimated more than 100 patients worldwide have been identified. DOCK8 protein helps maintain the structure and integrity of T cells and natural killer cells. It also aids in the movement of these immune system cells to sites of infection, particularly the skin. This is why DOCK8 deficiency patients have various immunologic abnormalities, including decreased T cell numbers and T cell function. Many patients with autosomal recessive hyper IgE were from consanguineous parents, which essentially means that the parents were close relatives. Hence, they had a higher chance than unrelated parents to carry the same abnormal gene. This increased the risk to have children with a recessive genetic disorder. Lastly, defects in TYK2 also lead to impaired Th17 function. TYK2 is also involved in signaling for type 1 interferons, which explains the increased susceptibility to viral diseases of autosomal recessive hyper IgE patients, and also interleukin 12, which explains the susceptibility to mycobacteria and salmonella.
The diagnosis of hyper IgE syndrome is suspected based on clinical symptoms such as frequent skin infections, abscesses in the skin or lungs that reoccur, eczema, which is a condition of dry and itchy skin, as well as recurrent pneumonia. Firstly, NIH hyper IgE scoring system should be used, as this system allows us to quantify the laboratory results and clinical findings and provide us with a classic autosomal dominant HIES patients often have scores above 40. Autosomal recessive HIES patients will have a significantly lower score given the absence of pathologic fractures, scoliosis, hyperextensible joints, and pneumatocyst formation. The presence of cutaneous viral infections appears to be a key feature that helps to distinguish DOCK8 deficiency from STAT3 deficiency. Secondly, the diagnosis is then further supported by taking a patient's CBC with differential and serum IgE levels, amongst other tests mentioned below. Current methods to measure IgE including assaying serum IgE levels by standard ELISA or by assessing the intensity of the allergic reaction via skin prick test or a radioallergosorbent test, which is also known as RAST. Elevated levels of eosinophils, known as eosinophilia, and elevated IgE levels along with other characteristic symptoms mentioned earlier can be a strong basis for diagnosis of hyper IgE syndrome. Serum IgE is often above 2000 IU per ml and can be as high as 50,000 IU per ml. Decreased T cell, B cell, and natural killer cell numbers have also been described in DOCK8 deficient HIES patients, which are otherwise normal in mutated STAT3 HIES patients. Lastly, genetic testing for gene DOCK8, PGM3, SPINK5, STAT3 can identify the gene mutations and is done mainly to confirm the diagnosis or to help predict inheritance patterns. STAT3 testing is now commercially available by a company called GeneDx. However, DOCK8 and TYK2 mutation testing is only available through specialized research centers.
There's no permanent cure, but only treatments like using antimicrobial drugs. Prophylactic anti-Staphylococcus antibiotics are used to treat eczematic rashes due to Staphylococcus aureus infections. And to treat infections caused by Candida species like Candidiasis, fluconazole is the best to opt for. Skin hydration, topical corticosteroid and moisturizing cream can make the eczematous dermatitis better. Diphenylhydramine or longer-acting antihistamines such as loratadine and cetirizine can be used to treat pruritus. Since pneumonia is a result of Staph aureus infection, hence it can be treated with intravenous neficillin or vancomycin for methicillin-resistant Staph aureus strains. For other type of strains, intravenous cefuroxime can be used. Superinfections by Aspergillus species and lung abscesses and Pseudomonas aeruginosa or other gram-negative bacteria require intravenous amphotericin for several months and combination of anti-pseudomonal beta-lactam aminoglycoside besides surgical interventions that is necessary. Bone marrow transplantation is curative for some primary immunodeficiency diseases like DOC8 deficiency with its role crucial in T and B cell development. Hence, bone marrow transplantation at an earlier age is required, yet depending on the penetrance. As mentioned earlier, surgical intervention may be needed to drain abscesses, emphysemas, and to treat bronchial artery bleeding. Lobectomy can also be done to remove the part of diseased lungs, but it may be technically difficult. Procedures like bracing and spinal fusion surgery can be performed to treat scoliosis, and fixation with screws, plates, and rods can be done to treat fractures. To wrap it all up, hyper-IgE syndrome is classified into two depending on the mode of inheritance, where autosomal dominant involves STAT3 gene mutation, whereas autosomal recessive involves not just mutation at TYK and DOC8 genes, but also by reasons of unknown mutations. On top of that, both type differs in terms of immunologic phenotypes, where the recessive type favors susceptibility to neurologic and autoimmune complications and lack in skeletal and dental abnormalities that's otherwise exhibited in dominant type. On a good note, in year 2016, a study has been started for vaccine using recombinant proteins by the National Institute of Health in the US to protect autosomal dominant hyper-IgE patients from Candida and Staph aureus infections. So hopefully, there will be a significant cure for this disease in near future.
Thank you so much for watching. We hope you enjoyed the video and learned something new today.